Sometimes, despite our best efforts at proofreading and complementary base pairing, mistakes happen that cause changes in the DNA called mutations. So, a mutation is just any change in the genetic information. And mutations are usually given a bad rap, but they could really be neutral. So, mutations could be beneficial if they cause a change that makes you more sexually fit, reproductively fit, or they can be detrimental if they make a change that causes damage. Mutations can also be neutral if they don't cause any noticeable functional change. That mutation just persists and doesn't cause any harm or good. So there are several different types of mutations. A silent mutation is going to be a mutation that does not affect the protein product. So this means even though a piece of DNA was changed, the original protein is going to be the same as the mutated protein, because this usually happens when the mutation is in the third amino acid of the codon. Sorry, the third nucleic acid of the codon. Because there are a lot of different codons that code for the same amino acid. So if you switch between two codons that code for the same amino acid, it's not going to change the overall protein. A missense mutation is when the resulting protein only changes by one single amino acid. So in the picture on the right, you can see an example of a missense. So the original non-mutated protein is supposed to read met, lyse, phi, gly, and alla. However, there's a change where the C got changed to a T, and the resulting mRNA has an A now instead of a G. And so in this one spot, it's going to code for a different amino acid. So instead of glycine, we're going to get serine. The rest of the protein is still the same, except for this one amino acid. Sometimes this is really bad. In the case of, for example, hemoglobin, this causes sickle cell anemia. However, if the amino acid that's changed is very similar to the original, it might still result in a functional protein. A nonsense mutation, we'll think about what the word nonsense means. It means crazy. Is going to be when an amino acid gets changed into a stop codon. So this is going to dramatically change the shape of the protein that's made. Imagine, for example, that this third phenylalanine, instead of coding for phenylalanine, coded for stop. Well, then the amino acid chain would stop after two amino acids, and that would not work. A frame shift mutation occurs when a nucleotide is inserted or deleted. So you can see two examples on the bottom. We have a deletion and an insertion. And this causes an issue with the reading frame. And by the reading frame, I mean how the codons are read. So in the deletion, the G that was in the fourth codon here is going to become the third amino acid of the third codon. And now the codons are off by one. The same thing happens down here. The third codon was supposed to be UUU. Well, now it's UUG with this G inserted. And every codon after that is going to become different. So a frame shift mutation usually causes a really big change to the amino acids, to the protein, where every single codon after the mutation is going to show a different amino acid than it was supposed to be originally.